Bam.
We're not going to sit here and take a bunch of notes. You should have already done that. So this is more you just looking at it, going, wait, what's this, and that kind of thing. Okay? If you want to sit down and take a bunch of notes and that kind of thing, well, we got it on tape. You can take your time and do that later if you'd like. Okay? Just see if you can follow this. This is the thylakoid membrane. Where is the thylakoid membrane located?
Chloroplast.
In a chloroplast. Where is the chloroplast located?
Inside a plant cell.
In a plant cell. And where's the plant cell located?
In a plant.
Plant. We know where we are
Step
now?
one.
They're usually in the leaves of plants. There's also some of these in the stems. But usually you find the chloroplast in the leaves. And so what we're trying to do here is photosynthesis. Do y'all remember the photosynthesis equation?
Yes.
CO2
6 CO2.
6 CO2.
plus 6H2O
6 H2O.
Get a
yields
zoom in on that.
6O2
Sorry.
C6H12O6 plus 6O2. And there's got to be some light coming in somewhere. So you can put light in the front here if you want it.
or on the arrow.
Or on the arrow, yeah, sometimes you see it there.
Okay.
And so,
Sorry, I got something that's supposed to be class.
What was the point of that?
okay, so
That's Mac, by the way.
you ought to be able to see where the carbon dioxide comes in, and the water comes in, and the glucose comes out, and the oxygen comes out. You ought to be able to see that as I explain this. So we'll refer back to this equation a bit. So, here we are. This is the membrane of the thylakoid membrane, these little tiny circles in here. And in your book, they blew it up real big so you could see. See the thylakoid membrane there? And that is right here, represented right here. Okay? So, what happens is light hits the chlorophyll, and the chlorophyll absorbs the light, and the electrons jump up to a higher energy level when they absorb the light. And I got some electrons here, and they'll jump out of the chlorophyll, and then they'll start moving down these proteins. These proteins are called the electron transport chain. Yes?
I don't really I get that energy is like electrons, I guess. Like, how does that even work?
Well, it's a physics thing. Science don't understand it fully, but somehow the electrons absorb the light,
They call those.
and it makes them more energetic.
And the electrons are sitting on the
The electrons are in the chlorophyll, and the chlorophyll's on the thylakoid.
I've got a question.
Yes.
So if they wanted to invent a machine that made food out of sunlight, wouldn't they just smash up a bunch of plants, take the chloroplast, put them in one thing, and then bam, you got a food-making machine?
You can do that.
It's called a farm.
Yeah.
Yes, but there's a machine that does it.
Well, you can work on that.
Just like straight up piece of sugar.
Okay.
Yeah. Okay, so the electrons jump out
Farms aren't
after
machines.
absorbing the sunlight, and they start moving down the chain. Then, they're energetic, and their energy is used by these proteins here to pump hydrogen ions into this area here that's called the thylakoid space. Let's write that down. This area is called the thylakoid space. You know what you call the area outside the thylakoid membrane out here?
Not the thylakoid state.
It's the stroma.
Oh, that was my second guess.
So as the electrons move down the chain, some of their energy is used to pump hydrogens in.
And the hydrogens that are sitting there. In
The hydrogens that are sitting
And
all
those
around.
hydrogens are pumped through a certain something called the...
What's that?
What's the thing that the hydrogens go through?
Oh, it's a protein here. Some protein in the membrane. Don't worry about it. Okay?
Okay.
Now, the chlorophyll is now missing some electrons.
Uh-oh.
And so any water that happens by, the chlorophyll will steal electrons from the water. And that breaks the water molecule apart. Because those electrons were being used to share with the hydrogens, between the hydrogen and the oxygens, as a bond to hold the hydrogen and oxygen together. But when those electrons leave, the water falls apart. Okay? You good, Savannah?
Good.
Can you understand what you had sideways?
Yeah.
Okay. Yeah.
Is there only one of these little chains? Because like if you look at like that's only like on this much of the thylakoid thing. Like, is there only one of them?
No. Remember, the thylakoid is a three-dimensional structure that comes out like this, and so these things will be all along the top and the bottom,
so it's not
and the
just like,
sides.
Okay.
They're just everywhere.
One for clarity.
Okay.
And so this just continues to happen as long as the sun's out. Sun hits the chlorophyll, electrons go down the chain, hydrogens get pumped in, water gets broken apart, and it just continues to happen over and over. A lot faster than I can move things around with my hands. Okay? Once two waters have been torn apart, the two oxygens will stick together. And now you have O2. You've created oxygen. And the oxygen simply floats right through the membrane and floats out of the cell, out of the chloroplast, and out of the cell, and eventually out of the plant. And that's, so we've covered this now. We've covered where the water comes in and where the oxygen is made. Do you see? Yes?
So hydrogen comes from two places. It's pumped in and it's from the water.
It's pumped in and it's from the water. And so this is an important point. We build up a lot of hydrogen on the inside right here.
Acidic.
Yes, it's very acidic, very good. That's how they discovered this was even happening, by testing the acidity on either side. Now, if we have a lot of hydrogen on the inside, and remember, a hydrogen ion is also called a proton. So if we have a lot of protons on the inside, and not very many on the outside of the membrane, they're going to want to get out.
Concentration.
We have a concentration gradient, and the hydrogen ions will go out. There's only one way out, through the ATP synthase protein. And when the hydrogens come out, ADP and P are put together. And that will happen over and over. Hydrogen comes out, ADP and P are put together.
Now, where's the sugar made?
We're getting to that. We haven't gotten to that yet. We're making ATP, though, aren't we?
Yeah.
Oh, wait, no, I know it is.
That ATP is going to be used to help make sugar.
Yeah. And those those things that are floating around right there, the carbon dioxides that have.
We're going to get to those in just a second.
Okay.
So it wants to go out because it wants to be for more hypotonic. It wants to go to hypotonic solution.
No, not hypotonic, hypertonic. If you have a high concentration on the inside and a low concentration on the outside, they're naturally going to want to go out just because they're moving and they want to spread out. And so that's just diffusion. And we call that chemiosmosis. Movement of chemicals from one place, from high to low concentrations. Chemiosmosis. Yes.
So are H plus ions, are they a gas in this state, or does it really matter?
They're protons. That's a good question. Yeah, I guess they would be. This is all, I don't know.
Yeah, it doesn't really matter. Okay.
Good question. No, it doesn't really matter. Let's look back at the electrons that were moving down the chain. What happens to them? Well, they go into photosystem 1 chlorophyll, and then sunlight hits that chlorophyll, and they jump out again, and they keep moving, and they eventually come to the end of the chain. And at the end of the chain is this special molecule called NADP. And it picks up the electrons. And it'll also pick up a hydrogen ion when it does that. A proton. It picks up electrons and a proton.
And then you've got NADPH.
And now you have NADPH. And that's another thing that's made in the light reactions. Electrons come down the chain, picked up by NADP and an H, and now you have NADPH. So the light reactions use water up, use light, make oxygen, make ATP, and make NADPH. The ATP and NADPH are going to be used in just a second to make sugar. Yes?
I actually need to go, so I need one.
Okay.
Need one last thing. Do we need to know about the 3GP, the GP3, and all that stuff, or do we just need to know basically how?
That's part of this.
Okay.
Yeah.
So we need to know the order.
Give me a video.
I'll give you.
Yeah, you don't need to know. You don't need to know all of those parts, but you need to know some of them.
Okay.
And that's what's in the dark reactions that I'm doing right now.
Calvin's. All right.
The Calvin cycle. Yeah.
All right. Thank you very
Okay.
much.
Can you watch the Calvin cycle?
Yeah.
And then I'll put it on the computer right now after y'all leave.
Really?
Okay. You want to leave too? Okay.
Yeah.
Bye.
All right. See you later.
Okay.
Thank you so much. Appreciate it.
You're welcome.
Then there was
And then there were 3.
Hello.
This is our APBio class right now. You should kick everyone else
Y'all
out.
would be guaranteed 5. Okay, so now we get, this is called the Calvin cycle. It used to be called the dark reactions. All these little blue things you see here are carbon dioxide. And what happens, and in the book, it shows it. It shows this picture in the book. Whoops. Shows this picture in the book to explain the Calvin cycle. And this is what I was saying. You don't have to know every little step. That's too much to know. There's only a few steps that are important. The first step is all these molecules, these are carbon dioxide that's floating around. They came in from outside. Carbon dioxide just floating around in the air, right? And the carbon dioxide comes in, and the carbon dioxide molecules will stick to these molecules that are already in the stroma. These molecules have a name. They're called RUBP, which stands for ribulose biphosphate. And they show them up there, right there, RUBP. And you see it says C5, that means it's a 5-carbon molecule. Now you have to imagine there's other stuff on here too, other than just carbon. There's oxygen, there's hydrogen, there's phosphorus. But I can't show you all that because it gets too confusing. So I'm just going to show you the carbons that are involved. These molecules are CO2. So there's actually oxygens stuck to these molecules as well. But I can't show you that because it gets too confusing. So we're just going to worry about the carbons here. Are y'all good with that? So, in this first step, carbon dioxide comes in and attaches to the RUBP. Click, click, click. Now we have 6 carbon molecules. 1, 2, 3, 4, 5, 6 carbon molecules. The 6 carbon molecules, no longer called RUBP, immediately break down into 3 carbon molecules. These 3 carbon molecules are what are acted upon by ATP and NADPH. Those 2 things that were made in the light reactions come in here and change these things around by adding energy and adding protons and adding electrons to them. And then these things just go back to where they came from. The NADPs will come over here and collect more protons and electrons. And the ADPs and Ps will go back here to get put back together.
So those are the taxis.
They're just taxis, yeah. These brought over energy, and these brought over protons and electrons. And these are now new molecules. I'm going to move these protons and electrons, but you just have to imagine they're still in these molecules. And now these molecules have a name. We call them
Pyruvate.
G3P.
Right.
Pyruvate's that other thing.
Pyruvate's over there. We'll
Yeah.
get pyruvate in a second.
When they break apart.
One of the G3Ps is stored away. The other 5 G3Ps are made back into the RUBP that we started with.
Then it happens again, and then it makes glucose. Yes.
Happens.
And ATP is more ATP is used to turn those things back to RUBP. So ATP is used again in that step to change those things back to what they started with. So, just like you said, it happens again. CO2s come in, they attach,
Breaks apart.
these break down a little bit, NADPH and ATP come in, change these around. Now it's called what?
G3P.
G3P. One of the G3Ps is stored away. The other 5 G3Ps are remade back into RUBP. The 2 G3Ps that were stored away bond together, and that's glucose.
6 carbons.
So we've made, it has 6 carbons, C6H12O6. So we've made our glucose from our CO2. That's photosynthesis. In 18 minutes.
Yeah, not that hard, probably.
I had more trouble with that one. That one.
Let's. You want to do this one now?
Yeah.
Okay.
We move this podium.
I'll move the podium. Silly. I'm being silly, y'all. No, I don't want to save the changes. The next part is respiration.
And enable all content.
I hate having to do that.
Cool.
Usually they say, don't ask me this again, and I say, yes, never ask me that again. But they don't give me that choice here.
You know, if you go to your settings, you should be able to change it.
Oh, really?
Perhaps. I could do it for some extra.
Okay, so, yeah, you were going to change my
Yeah.
refresh rate, and you haven't done that yet. Anyway, so the glucose, let me change the way my glucose looks. The glucose is a high energy molecule. It's got all the energy from the sunlight stored in these bonds. And so it can be used. It can be broken apart. And what we have over here, this picture I have on the board here, is the mitochondrion. See this mitochondrion up here? I've kind of turned it sideways and shown it to you big right here, just the middle of it. You know what the middle of the mitochondrion is called?
The matrix.
The matrix.
Nailed it.
You know what this area between this inner membrane and the outer membrane is called?
Intermembrane space.
Nice.
It's on the other side.
Inner membrane. Oh, it's on the other side.
I didn't see it.
That's too easy. See? So this is the inner membrane, outer membrane, intermembrane space, matrix, and we call these folds cristae. Cristae. I remember Christy Barrow took this class and she loved that name. And so this glucose is going to cruise on over here, and we're going to tear apart the glucose now. That's what mitochondrions do. They take the glucose and they rip it apart to make ATP. And it happens in 4 steps. Do you remember the 4 steps?
Reduction? No.
Wait, are we talking about the
Glycolysis?
People
Green people,
Can't excel.
can't excel. Yeah.
Can't
But
excel.
with an E. Oh,
good
oh,
people
yeah.
can excel. Why green people?
It's something.
E.
I think of the piano. Every good boy does
No.
fine, but it doesn't really work.
What's the E? Accelerate?
Yeah.
Good
G-
people can
like,
excel.
Like Microsoft
So
Excel.
something with an L, A,
Like-
then with an E.
Oh.
So the first phase, G, that's glycolysis. That's when the glucose molecule is ripped in half, basically. That process is done by enzymes outside the mitochondria. We're not in the mitochondria yet.
That's right.
Outside the mitochondria, there are enzymes that break it in half. Now your book shows you this long process. Here it is. Your book shows you this long process by which it's broken in half. But I just want to do it short, don't y'all?
Yeah.
Boop. There you go. That actually happened in 8 steps, but I'll do it quick. And whenever you take a large molecule and you break its bonds, energy is released. And protons and electrons come flying off. So the energy is picked up by ADP and P. The energy is picked up and ATP is made during that breaking. And the electrons that come flying from the structure are picked up by NAD, an electron carrier. And the protons that come flying from the structure are picked up by NAD. And now we have a new structure called, a new molecule called, do you know what?
NADPH.
NADH.
H.
It's
Right.
not the P. NADP is in photosynthesis, so it's called NADH. So you will see that as we're going along in respiration, every time a molecule is broken apart a little bit, protons and electrons come flying off, and NADH is made. So that's the glycolysis. In glycolysis, we actually make 2 ATP and 2 NADH. And what are these called?
Pyruvate.
Pyruvate.
Here we go.
That was your molecule. Your molecule?
Hers.
But I nailed it.
Now the pyruvate goes into the mitochondrion. And in the mitochondrion, we go to the next thing. It's called the preparatory reaction. The preparatory reaction combines the 2 pyruvates with a special molecule called CoA. And when they combine,
Acetyl-CoA.
2 of the carbons come off. Just put it in the box.
Oh, yeah.
And we end up with acetyl-CoA and 2 carbon dioxides, which just float away.
That's the carbon dioxide. Some of the carbon dioxide.
That's some of the carbon dioxide given off in respiration. We should write down the respiration equation probably. You remember the respiration equation?
Okay.
Opposite.
C6.
Yeah, C2O6
Plus 6O2.
Arrow.
Yeah.
Energy.
6 CO2
Two
plus
plus
6
6.
H2O plus energy.
Okay. So you saw when our glucose came in, at the very beginning our glucose came in and got torn up. So we've already seen the glucose. Now we're seeing some of the CO2.
Third of it, right?
Yeah. Some of the CO2 comes off right here whoop, in the preparatory reaction. When these 2 pyruvates come in, they hit acetyl-CoA and they break off, and these 2 parts float away as carbon dioxide. Remember, there were oxygens already stuck to the structure. And so some of the oxygens come off with this. Okay? Now, whenever you take a large molecule and you break bonds, what happens?
Energy.
Stuff flies off.
Oh, that too, right?
Protons and electrons. And so here we go, some protons and electrons flying off the structure. NAD picks up the hydrogens, picks up the electrons, and we form more NADH at this step. The NADH is just going to slide right over here. And we're going to see it again in the electron transport chain in just a second. So in the preparatory reactions, we make 2 NADH.
What are you doing?
Is it hard to do?
Don't worry, I got this.
So there go the carbon dioxide floating away. Now, what's the next step?
C.
What's the C stand for?
Citric acid cycle.
Nice.
That's it.
Citric acid cycle.
Yeah.
What happens in the citric acid cycle is this acetyl-CoA is completely broken down.
Oh yeah. It's completely.
It happens in a bunch of steps. And they show this in the book. But I don't care that you know them.
Okay.
Here's the picture in the book for those of you at home. This picture is in your book explaining the citric acid cycle. But all that's happening really, is this acetyl-CoA that we produced in the preparatory reaction is just getting torn apart by special enzymes and molecules that exist in the matrix. And so it shows you step by step how they get torn apart. And it's a cycle that can happen over and over again. The thing that's tearing these apart can do it over and over again. But what happens when you break apart a large molecule?
Stuff flies off.
Stuff flies off of it.
Nailed it.
Protons and electrons. And these protons that come flying off the structures are picked up by these electron carriers.
They carried over there, and then they go outside.
And the electrons that come flying off the structure are picked up by these carriers. The electrons that come flying off are picked up by these carriers.
Is this after acetyl-CoA has been torn apart or no?
Yeah.
Yes, this is acetyl-CoA gets torn apart,
And
those protons and electrons come flying off and get picked up by the carriers.
Is that what it is?
This is while it's being torn apart.
Okay.
And so these carriers are called NADH, which we've already seen, and another one's called FADH. It's actually called FADH2 because it picks up 2 hydrogens. We haven't seen that yet, but it's just another electron carrier. It's another taxi. So the citric acid cycle is going to end up making 6 NADH, 2 FADH2, and 2 ATP are made in that step.
Aren't like all the ATP made into E?
Well, there's 2 ATP
Well, I know,
made
but it's
here,
like
and there's 2
4.
here, and there's going to be like 32 made
Okay.
here.
Yeah, because it's like 32.
Yeah. Let's look at the final thing, electron transport chain, and you can go home.
Sweet.
Here's the electron transport chain. Right on the Christie, on the inner membrane here. And what happens is, I'll bring in this NADH also, that we formed earlier. All these electron carriers simply come to this electron transport chain on the membrane of the mitochondrion, and they drop off their electrons. And when they do that, the proton falls off too. And the electrons start moving down the chain, and the hydrogens get pumped out by the energy supplied from the high energy electrons moving down the chain. It's the same thing we saw in photosynthesis. Photosynthesis had hydrogens getting pumped in. Respiration has hydrogens getting pumped out. But it's the same idea. So here we go again. NADH comes up, drops off electrons. Electrons move down the chain. Hydrogens get pumped out. FADH2 drops off electrons at a different spot in the chain. Drops off hydrogens. Electrons move down the chain. Hydrogens get pumped out. All this dropping off and moving of electrons and pumping of hydrogens makes it so there's a lot of hydrogens in the inner membrane space and not many hydrogens in the matrix. So what are the hydrogens going to want to do?
Follow their concentration.
Come back in.
They're going to want to come back into the matrix. Just like Neo. And so they come back in through this ATP synthase. And just like in photosynthesis, every time a proton comes back through, a DP and P are put together. So proton comes through, ADP and P are put together. We call this difference, it's called a concentration gradient. It's also called a proton gradient because these are protons. So, we make a bunch of ATP this way because these are going to keep coming and keep coming and keep coming as long as NAD is dropping off its electrons and the electrons are moving down. Hydrogens are going to keep coming in. And we make 32 ATP for every glucose that we tore apart. But we're not quite done. There's one more thing. Do you remember what it is? What haven't we seen in our
Water.
equation?
Oxygen.
We haven't seen oxygen, we haven't seen water.
High concentration or low concentration? Is that what happens? When is that?
Here's the oxygen that was produced by the plant. Well, it will float on over here. Float in the mitochondrion, come all the way to the end of the electron transport chain in the mitochondrion, and pick up the electrons. And when it does that, it breaks apart from the other oxygen, and it'll also pick up 2 hydrogens. And guess what we have now?
H2O.
Water. Bam! That's where the oxygen comes in and the water comes out. Oxygen is the final electron acceptor at the end of the electron transport chain and will pick up the electrons and hydrogens that come back in and form water. That water is called water of metabolism, and it's important for organisms. But oxygen has to be there or these electrons will have no place to go and the whole thing will stop. If you can't breathe, there's not oxygen picking up these electrons and your cells can't make any ATP. And you'll die. That's ultimately why you die when you're drowning or when somebody's suffocating you. Is your oxygen is not getting here to pick up these electrons and your cells can't make ATP and then they can't work. They can't do their job.
And that's what causes fermentation. It needs.
Yeah, it'll go to fermentation first. Yeah.
I see.
Yeah.
Oh.
So y'all are out of here, huh?
Oh.
I hope you got that. See ya. Thank you. Good job. Do y'all